It's a great pleasure to have Michele here. Michele who is in Oxford University. For those who don't know, in the UK, there is often said to be a great deal of competition between Oxford and Cambridge. They race in boats, they play rugby, they do football. But actually, in terms of research, we have fantastic links between the two centres. So it's wonderful to have you here, Michele. Thank you very much for doing your blog in the past. But what I thought I would do is just start off by perhaps introducing us. So, what are you in Oxford University? And then let's go back to your early life after that. So, what is your position within the university?
Um, so within the university, and thanks for having me and inviting me, Roger and Ellie. I'm a professor of clinical neuroscience, so that just means patient-facing research on the brain and spine. I'm sort of a member of quite a large department, but I'm also quite defined by working in the NHS, which is the healthcare system in the UK for the last 32 years, and being consultant neurologist coming up to my 20th year now, particularly focusing on Parkinson's. So
Yeah.
yeah, the years are going
So,
quick.
so your job, which we'll come on to discuss later, is not
Mm,
only research, but you offer a service because the NHS is obviously the standard of care where
yeah.
anyone with a medical problem, or in this case, neurological problem, would come in. So you're a bit of an unusual sort of individual
Yeah.
because you offer a service and
Yeah.
you do research, which in the UK is a little different because unlike in America where it's much more common, I would say
Mm,
it's less common here. Um,
mm.
so Michele, how did you get into medicine? So how did you begin in life? Do you come from a family where every single person has been a doctor for generation after generation and you just
No,
followed?
no. So I'm the only one in my family when I got into medical school. So my mother was a nurse. So I guess there's a little bit of caring thing going on there. But my grandparents, so my name, I've got a Chinese father who, so Doctor Who was my first ward round with, if anyone's, a big scarf. In those days, it was Tom, the actor playing the Doctor Who series.
Yeah, which
So
is a big thing
that's
here in
why
the
I have a
UK.
China.
I don't know how much it's international, but I don't know if it's known in the
Yeah, it
States, but
might
it's
be
a
a
very
bit
big thing in the
specific.
UK. Doctor Who is a very
Tom Baker,
popular.
yeah.
Yeah.
And my mother's German, but they met in Oxford, funnily enough, when my dad was a student doing economics and my mother was a nurse learning German and seeing the world for a year. So yeah, how did I get into medicine? I guess actually slightly by accident. I was going to do law. It was either law or medicine because I've always liked a good argument. And I had to kind of get my dad to sign off my in the UK, when you're 16, you have to sort of specialize in three or four subjects. And I kind of was going to do French history and English because I love writing. And then my dad went, well, when do you think you do medicine? I was like, well, I'm not sure now. And he was like, just do physics, chemistry, and biology. Keep your options open because you can still do law later. Really weird. I hadn't really thought about it. I ended up doing it. And then yeah, kind of thinking I could probably do medicine because there's enough scope in here for me not to get bored. And
Yeah.
applying for medical school was a bit of a joke because I applied all mainly to London. And at this time, people in the US won't know, but there was quite a kickback of people with ethnic minority representation in universities. And I got an unconditional offer from St George's Hospital for a place
In
at medicine.
in
Or it's
London.
sort of very, yeah, very low grade. And everyone was laughing at all my other interviews because they said, of course, with your surname being who and George's, it was all in the news because a journalist had applied with two identical CVs to George's, one with an ethnic minority surname and one with a white sort of Caucasian one. And of course, as predicted, the white Caucasian got this offer, and then it was a big scandal that they were trying to address. So I ended up going to King's College in London. I really liked the idea that it was large. I wanted to go to a university of life, not just a medical school, and it had a lot of international students, and I had a great time.
So you did your preclinical and your clinical in London.
Correct. Yeah.
Yeah. And I mean that's a few years ago and you're still
Mm.
hooked on medicine. You're not going to switch to law.
Not then, no. Yeah, I had good.
So
Yeah,
so you went through
yeah.
your medical school and for those who don't know it, you do your preclinical, do your clinical, and you then come out of it and you're probably part of the old system I was you then have to do general medical training. So how did you get into
Mm.
neurology and then specifically into Parkinson's?
Yeah, so as a medical student, you, as you say, you rotate through different specialties, and I was quite sort of unconvinced as to what I wanted to do. I think the
Yeah.
number one thing was I wasn't going to do surgery or be a general physician, general
Okay.
practitioner, or family doctor, I guess they'd be called in the US. So I
Yeah.
knew I wanted to do something in medicine. I was really attracted to endocrinology because I think the systems of hormonal signaling and the gut brain and everything. And then I had some really inspiring neurologist at King's who basically taught us as medical students neurology. And this was at the old map of the house sort of unit. And one of them was Professor Nigel Lee, who is
Um,
really unusual in that he's an excellent clinician and also a fantastic researcher into ALS, MND. There was
Lou
Damien
Gehrig
Wren.
disease.
Yes, Lou Gehrig disease to be
Yeah.
the culturally specific. Yeah. And then, yeah, there was Damien Wren, who was the senior registrar, so not yet fully trained, and took an interest in me and encouraged me to write an essay which got awarded for Multiple Sclerosis Society. And then there were people doing Parkinson's, and I was involved in the deep brain stimulation
Was
surgical
this all during
programming
your this was all during your
as a
medical
medical
training?
student. I sort of was helping out, going to theatre, but not really doing too much, but interested. And then
So
after I'd
Michelle,
done two,
this was all whilst you were doing a medical student. It wasn't after you'd
well,
qualified.
yeah, I was, yeah, yeah. I mean, these were
Yeah.
the olden days.
Yeah.
So
Yeah.
as a medical student, you were actually really involved on the team
Yeah.
ward system, and you knew the patients, you would help out with blood assessments, clinical, it wasn't phlebotomist. And I could do cannula. So cannula is quite a big skill that the nurses like me for.
Yeah.
And then after I'd done, so I thought I could probably do neurology, but let's just try it as a sort of specialty as a 6 month job. So I did that and I was like, yeah, I love this. Of course,
So where
hadn't
did you
been
do that? Where did you
organised.
do your 6 month
So I did
job?
that at King's and I was working
Okay.
with Professor Ray Chowdhury. I was with Chris Clough. And I was also covering on call for neurology registrar. I was going out to Lewisham, which is southeast London near Brixton, big
Yeah.
African Caribbean minority population there, and I was doing clinics there. And I then thought, actually, I could do Parkinson's because, you form a relationship with the patient, you get to know them really well, and it's got therapeutic treatment options,
Yeah.
absolutely things that you can do for patients. And
So this was all
in
during your general
contrast,
medical training and your neurology
yeah,
training. You sort of came to
in
the
my neurology
realization as
bit,
you get towards the end that I could be a neurologist and I could be a
yes.
neurologist in Parkinson's and related
Mm,
disorders. So
that's it.
how did you transition into that specialty then? I mean, obviously you have to have applied for a neurology post, but how did you take that forward, wanting to do Parkinson's
Yeah.
research and
So I hadn't really planned it, but Professor Ray Chowdhury, who's very well known in the non-motor symptom world of Parkinson's, sort of offered me a research post. And it was sort of through a pharmaceutical trial. And I kind of hadn't really thought anyone would want to fund me. And I hadn't looked around. So I just thought that was brilliant and said yes. And then I did start off on doing the, in fact, in those days, it was the entacapone trial that
okay.
we were running, the first ever entacapone trial, placebo controlled, and we were giving
So
patients
this is a
that.
drug, for those who are not aware of it, this is a drug which you add on to Parkinson's. So it's what they call a
Mm,
COMT inhibitor and it makes the dopamine go further. So it's a way of trying to enhance the efficacy of the
yeah,
drugs. And this was the first ever trial to see whether
yeah,
it
pretty
really worked.
much.
Yeah.
Because this was back in about 2007,
Yeah.
2005, 2006. And the experience that we had was that patients who were fluctuating and wearing off, there was no advice to reduce their overall amount of dopamine. So we were adding in this entacapone and they were all getting dyskinetic. Well, a significant proportion were wriggling
Yeah.
around and they'd never
Mm.
had them before. There were 3 or 4 patients that kind of got this for the first ever time and a few more that yeah had had it before but it got a lot worse, and so that was a bit of a negative experience of a drug trial, because when we then reduced the doses we couldn't get rid of the dyskinesia, so
Okay.
it was learning about the patient journey and the motor fluctuations,
So that was your
and then
first
after
exposure
that,
to research was really a drug
yeah,
trial. Yeah. Yeah.
absolutely,
But you
and then
weren't put
after
off by the
that,
fact that the trial had shown things that were a little, I don't know if they
no,
were
no, not
unexpected, but
really.
yeah.
No, and at the same time, I was working at the Hammersmith. So the Hammersmith Hospital in London in those days, and it still is well known for brain imaging. and using particularly PET brain scans, but also MRI. And I started to get really interested in how you could correlate the patient in front of you and their performance on cognitive or motor tasks with what you were measuring in the brain. And
So
that led
just
to me
those two
then,
types of scans. So MRI scan
yeah,
is a structural scan, although you can see activation when people are doing tasks. And a PET
that's
scan is when you
right.
look at chemicals in the brain, like
You can look at
dopamine and such like.
chemicals.
Yeah.
And we were measuring dopamine levels in the brain, and we were
Yeah.
measuring glucose consumption of the brain. And we were also measuring effectively mitochondrial function in the brain using phosphorus MR spectroscopy, which allows
So
you
this
to
is the
measure
energy
the
center of the
energy
cell.
production, in
Yeah.
each brain unit, which is ATP, as well as phosphocreatine and other things. And I was lucky enough to then get a fellowship from Action Research, which funded me for 3 years to then do a proper PhD research. And it was focusing on brain imaging and also looking at clinical phenotyping. So it started off my lifelong interest really in patient subtyping and whether that links to biomarkers or other things that we can
Okay.
measure, and how relevant is that for the individual living with their Parkinson's.
So what came out of that, Michelle? So what was the main discovery of your PhD in terms of, different types of bug disease and how that matched to
Yeah.
imaging? Was there some sort of major
Yeah. Well, there were
conclusion, or
two.
did you just come to the conclusion it was all rather complicated?
No, there were two sort of things. One was that if you just measure glucose activation, many patients with Parkinson's already have significant reductions in their ability of their brain regions to use glucose. And we were able to show that correlated really well with cognitive function. So I was learning how to do 2 hour neuropsychological assessments with the poor patients, like
Yeah.
quite a lot for them to do. I was correlating that with the glucose, but I was
So,
also
so just so we got the
correlating it.
glucose. So the glucose measures
Yeah,
basically metabolism activity in the brain. So if your
metabolism.
glucose levels are down on the scan, that bit of your brain
Mm,
isn't so active. So
yeah,
it's rather struggling to be
that's right.
activated to do something. Yeah.
Yeah. And we were showing that the areas in the back of the brain, which is the temporoparietal cortex, were
Yeah.
reduced in glucose, but also in ATP. So they were energy deficient.
Right.
And we were able to show this in people who haven't got dementia. So I was really excited in using it as a potential early marker for people who might be at future risk of
Okay.
dementia. And the study, we kind of had about relatively small numbers, sort of 25 patients, and we had a control group. And also, the other aspect of my PhD was looking at clinical phenotyping in patients with Indian or African Caribbean
Okay.
heritage, and looking to see, do they have a kind of clinical phenotype that's similar to what we recognize in white Caucasian people in the UK, and does it differ from people from their original country of origin? And
Yeah.
I was sort of clinically looking at them. I was looking at their levodopa benefit by giving them a test dose, and then I was measuring their dopamine and glucose. And the take home message is it's the same.
It's
It's
the
the
same.
same in imaging, it's the same on most clinical measures. But the main difference is that the ethnic minority people had higher side effects with the medication.
Okay.
So they're more likely to have nausea or
Did they
vomiting
respond
or
as well? Did they respond as well to the
when you did
medication?
the challenge,
Yeah.
in general, there was no significant differences in the group,
Yeah.
but there might be genetically driven ways, differences in how people metabolize the medication. That was what we wondered about.
Okay. So just to sort of so at this point in your career, you're now finishing your research. You've shown that ethnicity, whilst it has some subtler changes within Pugsy, fundamentally is the same globally, Parkinson's,
Yeah.
and that using imaging, which is perhaps not the most convenient sort of thing to do in a clinic, reveals that
Mm.
there are changes in the brain which may be useful as a marker for things going wrong further down the line, or for some of the changes you see in bug disease, which will become relevant because obviously with your current work, in some ways, it's an extension from that.
Mm.
So, so on the back of that, you then moved to Oxford, or when did you?
Yeah, no, it's a complicated thing. I sort of got married, and my husband, who's 3 years older than me, is a neurosurgeon.
Uh,
So we've got neurology and neurosurgeons. I don't know what it's like in the US, but in the UK, there's like a bit of a jokey thing between the two of you, rivalry, bit of like Mickey taking. And so I got I got married to Simon, who's 3 years older than me, and he wanted to specialize in pituitary brain surgery, which is very endocrine and is quite a sort of medical surgical specialty, I would
Yeah.
say, because you work with a lot of endocrinologists. And he got offered a job in Oxford at the stage where I still had about a year left of my training. And our second daughter, Isabel, had just been born when we moved there. So she was 2 weeks old, and my son Ethan was 2 years old. So we did everything you're not supposed to do: move house, have kids, and start a new job. And in the middle of all this, I was basically desperately trying to transfer my training for this last year
Okay.
to Oxford, which eventually happened.
That eventually happened. And so once you were in Oxford, and everything had settled down a bit. And for those who don't know, I mean, neurosurgery in the UK there aren't many neurologists, but there are even less neurosurgeons.
Mm,
And neurosurgeon working on the pituitary, probably there are, I don't know how many, there are probably no more than about a
not many.
dozen, I think, in the UK. But
Mm.
but so you were settling down there, and then it must have been around this time that the Discovery Award came along and sort of set
Oh,
things.
no, no, we've got loads more time to fill in. So,
Okay.
actually, as a woman, I sort of had put my career on hold really for my family, my husband. I was working part-time. I was doing 3 days a week, and I finally managed. I was told I was 87th on the waiting list to go part-time in Oxford. And I remember driving back to London before we'd moved, sort of 38 weeks pregnant, sort of like little tears coming down my face, thinking, well, that's it then.
Yeah.
How am I? And the person in charge of the deanery said, well, have you considered living in Tame? Because it's a very easy commute, commute to London, two children under three, etc.
Okay.
But then luckily, some of my part-time colleagues said, "Well, just get London Deanery to pay for your last year, like a transfer," and they
Right.
did. Joanna Ball said, "Yeah." So I turned up, and it's like you have to have me now.
Yeah.
And then Oxford sort of were a bit suspicious because I was not an Oxbridge undergraduate. I hadn't gone to Cambridge or Oxford.
Yeah.
I'd gone to King's College London. But then they cheekily put me on an on-call at the main JR, which is where I'm now physically.
Which is the
But in
old big
those
hospital in
days,
Oxford. Yeah.
yeah, but in the olden days, the sort of general neurology, plastics, ENT, neurosurgery was in a different building 3 miles
Yeah.
away, and it might have been a different planet because I was
So
basically the only
basically during
one.
this period of time you were struggling with this to get recognized for what you were doing, and essentially your trajectory was NHS, probably
Sticky.
research
I
had
basically,
sort of gone
yeah.
into the past, and you probably weren't going to do research, and then you got this
I got a
opportunity through.
consultant job. Yeah, I got a
Yeah.
consultant job at Milton Keynes, and thinking that I wasn't really sure how it was all going to work out, but I learned a lot there, and I spent 7 years there,
Yeah.
commuting from Oxford. But in parallel, the really encouraging thing was I applied to Parkinson's UK, and I kind of in my first year, I think everybody does this if you're in a new post. So it's not like I was replacing a really busy. I was actually just a little bit of time twiddling my thumb. I thought, well, what really matters to me is research, so I'll apply. And I got a tiny grant from Parkinson's UK, which in those days was under £20,000. And it
Yeah.
was to look at the phenotype of people living in Milton Keynes with Parkinson's in the community. I worked with a research nurse, Leslie, who stayed with me for a decade, and she went to all the family doctors in Milton Keynes and identified people with Parkinson's and looked at how many of them were known to the hospital or specialist. And it's very consistent because 90% of people in the NHS with Parkinson's, the community, see a specialist nurse or a doctor for their Parkinson's. And the 10% that don't, they're older, they're more likely to be more frail or in a nursing home. So getting back and forth to clinics is more difficult. But that
Okay.
started a research thing. And so after about 6 or 7 years of doing that and learning a lot, working at the coal face, and I was leading PD, set up botulinum toxin treatment, set up atypical PD. I got approached by Kevin Tolbert, who's the head of my department, saying, "Oh, we're putting in this grant as a centre. Would you like to apply?" He said something then. He said, "It's very unlikely we'll get funded, but you do need to think about it because if you do come on board and we really need a doctor, a clinician like you, it could change your life."
All
And it
right.
did.
Yeah.
Yeah, so
So that
we
was,
did
when
get
did that come
funding.
to pass then, Michelle? When
That
was it?
came in 2008. We wrote the application. So I was a consultant in 2005, and then in 2009 we got the award. In 2010 we started the research, and I was thrown in at the deep end. I'd never done any cohort research before. I'd
So
never
what's
set anything up.
so, so this was so you'd obviously done the study of Milton Keynes knocking on people's doors, but it was a
That
very
was
sort of
for,
low
yeah.
key, low intensity study in some ways. And now suddenly the main thrust of the work, if I remember correctly, was to go out and collect everyone with Parkinson's
Yeah.
basically across the county,
In a
and then
kind
just
of
follow them over time.
population.
Yeah,
Exactly right. 2.4 million population. So we
yeah,
were estimating 1,000 people with Parkinson's to 1,500. And we were doing clinics from Oxford out to about 11 district general hospitals. I had to
yeah,
generate funding, matched funding to actually sustain because we were underfunded massively.
yeah,
I had some very, very bright people working in the basic science of Parkinson's, both cell models, genetics, and also animal models.
yeah.
And I was leading this cohort program.
So the project was basically collect all these people with Parkinson's disease, follow them over time, dissect them by looking at them clinically and any other measures I could think of.
Mm,
See where, so coming back to the original question you asked about different types of Parkinson's disease, could I identify
yeah,
that at this population level? And then presumably in the lab, they were sort of taking cells from people, growing cells and
that's
saying, well,
right.
you tell us they're different clinically. And in the lab, we'll tell you they're different because their cells behave differently. They have a different sort of pathology, if you like, or a different mechanism that might be driving those
Exactly
different.
right. Mm.
And so that began, and that was obviously a fairly major undertaking. So obviously you'd have to
Yeah.
slightly come back on your general neurology because you've also got to then manage all this team. And so this work then began, as you say, about
Mm.
15 years ago now. I'm saying 10.
Yeah. Yeah.
And so as that work evolved, what were the sort of major discoveries that you made in those early days, and then it'd be interesting to see how it's transitioned into where you've concentrated now, which I would guess, I mean, you're
Mm.
very well known for a lot of work in Parkinson's, but perhaps best known for two things. One is people before they get Parkinson's, so-called prodromal Parkinson's, which we'll come on to, and
Yeah.
the other is the development of apps and other things for monitoring things. But at
That's
those
right.
early days, I mean, this is 2010, early part of that
Mm.
decade. It was more clinical. And what were the major sort of things you discovered,
Yeah.
apart from that it was rather
So the
difficult to
key,
run big cohort studies?
so the key learning, I mean, we set this up initially thinking that we would have an at-risk group of future Parkinson's, and we designed it thinking maybe we'll have siblings or patients or children of people that have a known gene
Yeah,
that we can test for in the lab in our kind of Parkinson patients. And actually, the experience we had, we did get about 100 people who were family first degree relatives. But when we followed them up, actually, very few of them actually got Parkinson's, which is good news for them.
yeah.
But they
So these are
didn't really
relatives who've got a known
progress.
genetic defect. So if I
No.
had Parkinson's, you know I've got a gene,
Mm.
and those siblings, those children, brothers,
So
whatever, they
the
were tested for the gene.
103
Yeah,
The 103 were roughly broken down into about 20 to 30 people who had a gene that we
yeah,
tested for, GBA and LRP2. We didn't
yeah,
have the capacity to test for all the known genes, but those are
yeah,
probably the two commoner ones. And then the remainder were really altruistic people who were very involved and wanted to know more and help their sibling or their mother or father. But when we talk to people who have a gene, actually, a bit like with Huntington's, a lot of unaffected relatives really didn't want to do a test or
um,
didn't really want to get involved in research. And so that was a good learning point for me because I realized probably about 3 years in that we needed to change focus of our
okay.
at-risk group or prodromal group to people with a sleep disorder. And I was
So
attending
the
meetings.
idea was so the prodromal group there would
Yeah,
be I'm going to go looking, I'm going to find people with
yeah.
genetic forms of Parkinson's
Mm,
and then going to grab their relatives on the grounds they carry the gene, they haven't got the
yeah.
condition, and I'll follow them and then they'll develop the condition. So my prodromal group will be genetic
Mm,
forms of Parkinson's. And you
yeah.
realize that that was a bit of a
Not going to
non-runner,
happen.
A, because they didn't really do Parkinson's, and secondly, people weren't very keen
Yeah,
on it. So,
that's
okay,
absolutely correct.
okay.
Absolutely right.
And
Yeah.
so in those early stages, and do put your questions in the chat if you have any questions for Michelle about any of this. In these early days, you must have just It must have been very revealing, though, about what Parkinson's does over time. So you must have got some insights
Yeah.
into why
We
some
got
people
some
develop
really,
major
yeah,
thinking problems, some fall, some people do very well.
yeah, yeah, that's right. So what we, being a clinician, we were collecting sort of 2 hours of clinical assessments for this patient, as well as biomarkers from blood, DNA. We were doing brain scans, MRI, measuring the dopamine as well in their brain through a DAT spec. And we're also doing lumbar punctures. So, that was voluntary, but
Yeah,
about 15% of people actually consented to have that done. So, and we were taking skin biopsies, which were being used to grow fibroblasts into stem cells, into brain cells as a model in a dish, right?
yeah,
So what I got really excited about was just that fact that if you put in all the metrics and you just told the computer to look at it and go through the data and see if it could form subtypes, that the data drove the research without a clinician mucking it up and saying, no, it has to be like this. So in
yeah,
medicine, we call that an a priori hypothesis. This was purely data driven, it was bottom up. And what we were able to show in the 1,000 people with Parkinson's we recruited and then replicate in another 2,000 people from the UK tracking was that there were at baseline roughly 4 subtypes of people clinically
yeah.
that we all recognize as doctors at the coalface. So one of them, about 20%, is the tremor dominant person who has often a unilateral tremor and they might have some mild stiffness or slowness. Their gait is pretty good and their non-motor symptoms like loss of sense of smell, sleep problems, depression, memory is pretty good. We also see a group at baseline who we've now looked at
And so with
who,
those group, Michelle, I mean, traditionally those people tend to do
yeah,
quite well. Uh,
yeah, they
I mean,
do.
I often say
So
to
we
people,
then
you'll
looked
do
at the
quite well if you have a
progression.
lot of tremor. The problem is it's not most responsive to
Yeah,
medication. So,
that's it. Absolutely right. So we showed they didn't respond to medication particularly well, but that didn't really matter because they were
no,
functioning really well and they maintained that level and they didn't progress very quickly.
yeah,
And then we
okay,
saw another subgroup, about 30% of people who you could tell very easily at baseline that these were patients who were much more affected at baseline, both in their motor symptoms, but their non-motor symptoms as well. And interestingly, they did have a higher rate of GBA, which is the common gene mutation. They also had a very different clinical profile where progressing faster, motor
okay.
and cognitive. And then we had another group
But that
that
second
was,
group, which are about a third of
mm,
people, they
yeah,
at presentation they just had more problems at presentation, and then they progressed
more
more
problems
quickly,
around the board.
and so
Yeah,
they ran into problems with major
yeah,
thinking
higher
problems.
rates.
Yeah, okay,
Exactly right. Yeah.
okay, okay,
Um, and then we had another group that were very driven by what we would call psychological measures of depression, anxiety, poor sleep, apathy,
okay.
and had more symmetrical motor Parkinson's, intermediate progression. And then there was a group that
And can I
just
just
looked,
ask on that front? I
yeah,
mean, obviously, one of the
mm,
problems with these studies is the only way you know someone's definitely got Parkinson's is sadly when they've died and you've looked at their brain. So
absolutely.
presumably, as much as you can be certain these people have Parkinson's, even though they're
Yeah.
slightly different. Because you could say if they're very symmetrical and they've got these other things, have they really got
Yeah,
Parkinson's?
absolutely. So we were following them all up in
Yeah,
person every 18 months until COVID hit. So most people have been seen about over 5 years from their
yeah,
diagnosis. And we were following and looking very carefully for what we call clinical features to suggest they got a different diagnosis to
yeah, yeah,
Parkinson's. So, what's your response to medication?
yeah,
Are you falling? What's your progression be like? What do you have an eye movement problem to suggest PSP,
yeah,
etc.? And now we have post-mortem donation. So we now have the first 20 brains of people who've subsequently died, donated
yeah,
their brain for research, and we're looking at how that marries up.
yeah.
And we were reclassifying people as well according to diagnostic
Okay.
criteria. So
But this third
approximately
group you had were
five,
largely, I mean, if you followed them
yeah,
for 5 years, presumably
yeah.
if they had one of these other
And they
Parkinson's
had longer
variants,
follow-up. Yeah.
they wouldn't have done so well.
Yeah,
So
I think it would have manifest by the 5 years
yeah, I think
from
so.
what we largely know, but we
Yeah.
can't exclude the possibility. And I think that's the
Yeah.
case for all cohort
So
studies.
you've got group one, which are tremor, do pretty well. Got group
Yeah,
two, have a lot more problems when
much
they present, who
worse in
progress very well.
motor, cognitive,
And then
non-motor
you have this
symptoms. Yeah,
intermediate group, which is
yeah,
dominated not so much by motor problems, but more
but more
motivational, apathy,
psychological
depression,
sleep.
mood
Yeah.
things. And then
And
the
then the
fourth
last
group,
group, they're so much better than everyone else. You wonder if they've got Parkinson's at all. They have a good benefit and they progress really slowly. And again, those are patients that we've seen. You kind of think,
Yeah,
oh, this, I've never seen them untreated, but they're doing really well. And they
yeah,
do, but they have like, they fulfill diagnostic criteria.
yeah,
So that's now what we've done is take that finding on a step further, and we've tried to look at what's the biological signal
yeah.
of these clinically driven phenotypes, and we found some quite interesting things. They have the worst at baseline, progresses more quickly. It has a pro-inflammatory signal on basic blood tests.
Okay.
It has higher spinal fluid seeding amplification assay
So
readout,
this alpha
um,
synuclein in the
which
CSF.
is
So
the
they have
new
more
sort of,
protein in the CSF.
yeah, it's got a
Associated.
higher rate of particular genetic
Mm.
causation. So maybe, 12% will have a GBA mutation.
So the
And
people who
then
are going
that,
to do less well, you can see
yeah,
clinically, and underpinning
yes,
that is they seem to have more inflammation, they seem to have a
yeah,
genetic predisposition,
yeah,
and then in the
or
fluid off the
higher
back,
chance. Yeah,
they have an alpha synuclein, the protein
yep.
associated with disease, which seems to be better at seeding, and whether that's more of it or a different type would be unclear.
Mm.
And of course, trying to disentangle that's a little
Trying to
unclear.
disentangle is very complicated.
Yeah,
Um,
and someone's
uh,
asked a question, which is, because we had Per Borghammer on in previous ones. Obviously,
mm,
there's this
yeah,
idea that there's a
yeah.
brain first and there's a gut first. So if you had to map yours on, and this will take us into the sort of prodromal, did you find that, for example, the ones who had a lot of psychological problems had
Mm.
more gut problems? The ones who did less well had less prodromal. Was there a clear divide on them, or did they all not really
It's
very
not
clear?
so straightforward. I mean, we don't
Okay,
have a kind of, so I think with the brain gut first, sort of brain first is really interesting. I think the proper archetypal person with Parkinson's who is gut for first is somebody who has RBD, and we know that's about, if you look at sort of screening questionnaires and then you do PSG, that will be around
so PSG is where
40%
you put people in a lab and
sleep
do their
study.
sleep overnight, yeah, and record.
Yeah.
Yeah.
So we reckon about 40% of PD patients at diagnosis have got RBD, and they're the absolute sort of gut first because they've got higher rates of autonomic problems,
Yeah.
suggesting peripheral heart and nervous system involvement, higher rates of constipation. They also have higher depression, anxiety, and cognitive scores.
Okay. So you've got these four subtypes. There's a question about age. Did you notice any major age
Yeah,
difference?
it was a big problem because the patients who presented worse and progressed worse in that subtype were about a decade older. So,
Okay.
age again is the strongest environmental risk factor,
Yeah.
both for
And it's
getting
always difficult with that
Parkinson's
because, of course, is it
and
age or
progressing.
is it as you get older, you get
Yeah,
other pathologies as well.
there
So
is
it's
that too.
yeah.
Mm.
Okay. So you've got these four types. You've started to
Yeah.
describe the biological basis for it, and then obviously it was validated with another big cohort in the
Mm.
UK. This big UK proband, I think it was called. And
Yeah.
so you obviously moving forward with this, obviously COVID comes along,
Mm.
doesn't help with follow up. But then you sort of slightly shifted gear and decided that you sort of cracked Parkinson's, and now you wanted to go for pre-Parkinson's and prodromal. So how did you get into that? We've had Ron Postuma on this before, who obviously set
Mm.
up a
Oh,
study
right. You've had all
years
the big
ago
names.
on RBD and
Yeah.
followed them over time. But you have sort
Yeah.
of come from Parkinson's and going backwards. So how did you get
That's
into
right.
prodromal Parkinson's
So
disease?
I
And
tell
what's
you,
the
it was a patient. So in my
right?
first year as a consultant in 2005 at Milton Keynes, there's no sleep unit, there's no sleep study.
Yeah,
It's just you. And in my first year as a consultant, the first patient that came in, it was triaged to neurology, and it was an absolute classic description of dream enactment behavior, shouting out, punching, waking his wife. So his wife dragged him up to clinic.
yeah,
He's giving me a black eye. I've fallen out of bed. If
yeah,
I wake him up, he just goes back to sleep. He doesn't remember anything the next day. And I said, well, okay, you've probably got RBD. Do you want to wait a year to be seen in a sleep clinic, another year to have the sleep test? No. I said, okay, do you want treatment? Yeah. So we gave him some clonazepam. I examined him. He was fine. I didn't at that time say, because it was my first patient, think a bit
yeah.
about the implications of this diagnosis. And he came back a year later. And I've got all the videos. I showed this at
Okay,
meeting.
yeah.
Mild tremor, and then he progressed, diagnosed Parkinson's, and I followed him up for 10 years. He's very, very typical for somebody with Parkinson's disease.
Okay, so this alerted you to the fact it's prodromal. So can I just
Yeah,
ask you as an aside, because I've never understood this. Why do people with RBD always have violent dreams? Why don't they ever
yeah.
imagine that they're swimming or lying on the
Yeah,
beach
it's
having a
not all
nice time?
violent. People have analysed the dream content.
Yeah,
There is a mixture. I think probably there's a bias in
yeah,
ascertainment and diagnosis towards
okay,
men rather than women. Although males to females, it's much more common in men than women, like Parkinson's, but to a
yeah,
much greater degree. So if you
okay,
look at sleep clinic ascertained cohorts of RBD, it's 9 to 1 male to female. And I think that's because men are more likely to be violent in their sleep and punch. I mean, call me, a feminist, but that is the case anecdotally, and it's
so what
also
happens
been
with women with
shown.
RBD?
Yeah, so I think they don't tend to punch out, and it's the movement, the dream enactment, the
Yeah,
violent thing that often your partner's got a black eye or fallen out of bed,
yeah,
or you've actually hurt yourself falling out of bed that often brings them to medical attention. We do know that, I mean, it's difficult to generalize, but there is a proportion of RBD. And I've seen videos where people are just sort of talking
okay.
and, they're just talking about what they've done that day and they're having these conversations with people and there's
Okay.
just an odd twitch. Um, I've seen a
No,
video
it's
of
because
somebody
it's not
who,
so florid. So it's when people get very violent,
yeah,
that's what people
I
remember.
wonder if that's it. And the
Yeah.
speech is never, you can never quite understand it. It's often very slurred. So you might be able to pick
Yeah.
out one word or two. But then I'm not a sleep specialist. But it does seem to be more of a tip of the iceberg effect in terms of what
Okay.
we see.
So, so you've had this sort of. I mean, I think this is very interesting because I think people in the clinic. I mean, we got into sleep in another condition, Hunter disease, and it all came
Mm,
from a patient telling me about their problems, and so
yeah,
that triggered a whole avenue of research. So you had this
absolutely.
individual sort of experience of it, and obviously,
Mm,
when you then came onto your discovery cohort, you realised that your
yeah,
prodromal Pogsy's cohort was never going to appear on the genetic. So you decided at that point, actually, what might be a smart thing to do would be to move into prodromal Pogsy's
yeah,
using REM sleep behavior disorder as the hook.
yeah,
So I would go after people with a REM sleep behavior disorder and
yeah,
follow them. Is that
yeah.
the thinking?
Absolutely right. And I think we recruited our first patient probably around 2013. And it made me kind of getting to know a lot of sleep specialists and understanding
Okay.
their language. Because sleep specialists in the UK, there's not
Because that's
many
where
that
they
are
end up, don't
duly,
they? I mean, traditionally they've always go to
yeah,
sleep clinics because it's a
they always
problem.
go to sleep specialists.
Yeah,
And, you have a few people who are duly trained in neurology and sleep specialty.
yeah,
I don't know, in the US, it's often, yeah, a mixture, but more predominant sleep specialists. And you'll have a sleep specialist who only looks at respiratory problems. So
yeah,
what they're doing is ruling out things like obstructive sleep apnea,
yeah. When you
and they
stop
don't
breathing.
feel that comfortable diagnosing RBD. But I was lucky enough to collaborate with people in the Sheffield Sleep Clinic and Cambridge.
Yeah,
Remember, I emailed you and said, would that be all right? Because I
yeah.
knew you were looking looking at sleep. And you said that was fine. So we got, so and then we've got the Oxford Sleep Clinic. So we've now got just under 300 people who've all had the sleep study diagnosis of RBD, and we've
So
been
these are
following them
300 people
after.
with RBD and nothing else particularly
Yeah, we call that isolated, but
at the point where they were recruited.
yeah, if you look hard enough, a lot of them have got subtle symptoms already, like loss of sense of smell and constipation. And what
Okay.
we've found following them up is that over time, they start to kind of get other non-major symptoms like orthostatic hypotension, so feeling dizzy when they get up. They're more likely actually to have higher rates of depression and anxiety and apathy and cognitive problems than an early Parkinson patient. So their non-major symptom burden is higher in the RBD, and they're very aware. It's not like a
And
asymptomatic
that's not
condition.
related to not sleeping well. So if you treat the RBD and put them back to sleep, those
Yeah,
symptoms
no,
continue.
no, it doesn't.
They're not,
Makes
no.
no difference. No.
So if these
Mm,
with these 300 people, I mean, one of the challenges I think is knowing when people are going to transition. And obviously it's never a
yeah,
sort of one day I was fine, the next day I'm not. So
yeah,
over with the 300 people you've seen, how many of them have transitioned into Parkinson's or something like
yeah.
Parkinson's over
Quite
what sort of
high.
period of time?
We literally have most months we'll have a few converters that we're seeing face to face by the neurologist in clinic. So it's the latest counts between 65 to 70.
So that
out
would
of
be
the
within five
original.
years of diagnosis or within 10,
It
roughly.
approximates to, it's absolutely bang on what Ron Postuma reported in his large analysis of about 1,300. So it's about an 8% per year conversion.
Yeah.
And
And do they
it's
all go to
important.
Parkinson's or do they, because there's a
No,
thought they go to
exactly.
multiple system atrophy, Parkinson's,
They
dementia,
all go
dementia.
to an alpha-synuclein disorder. That's the
Okay.
critical thing. It's a prodrome for alpha-synucleinopathy. Probably about 40% to 50% get Parkinson's disease. About 30% will get dementia with Lewy body. 10%, 5 to 10% will have MSA, and the
Yeah.
remainder will be a sort of unclassifiable form of dementia. So it's
Okay.
really important, this group, because they're also an early prodrome for dementia,
Okay.
and they could potentially for part of it early clinical trials designed to prevent that onset or modify or slow it down.
So this would be the sort of the ambition is if we can reliably identify, well, we can reliably identify them, but they have to come into the system first with RBD.
Yeah,
Then you can say this is when we should be targeting our therapies to prevent them going on to
absolutely.
develop whatever else they're going to develop around an
Mm.
alpha-synucleinopathy. And
Yeah.
um, so you're obviously following these people up now.
Mm.
To do all of these studies to diagnose, REM sleep behavior disorder, really, I mean, you can do it on a history, but ideally you need to come into a sleep laboratory, get
Yeah.
wired up to gazillion different leads. Amazingly, people sleep. If you've never seen what it looks like to be in a sleep laboratory, I cannot believe that people sleep in it, but they obviously do because they've done
They
for you.
literally stick on the electrodes with super glue, and I've
Yeah,
had it done myself. So it's like having a Rastafarian dreadlocks of super
yeah,
glue and wires. So it's impossible to move.
yeah,
It's very hard to roll over in bed. And of
yeah,
course, you're sleeping in a hospital environment,
yeah.
even though you might have your own room. So one of the things we got really
So
interested.
I guess where I was leading was obviously
Mm,
this is not great. So moving
no,
it, what you'd ideally like to do is stick it in someone's bed. Um, it,
exactly.
and so this, I guess, is what's led you into the sort of world of
Yeah,
apps and sort of
it's got me into a
remote
bit
monitoring.
the techie
Sorry?
digital space. So we've got
Yeah.
something called the Sleep Wearables Project, where we're recruiting people at the time of NHS sleep study, but they're also wearing alternative kit in parallel, and then we're testing them at home to see if the diagnosis is as good or nearly as good as the gold standard
Okay.
diagnosis. So
Yeah.
we've got a number of sleep kits. I will just say that an ActiWatch will not diagnose RBD reliably. The
No.
main problem is that distinguishing the movements and the sleep phase in REM sleep is very difficult because you
No.
need some form, usually of an EEG trace or a muscle trace. And it looks, somebody could just be lying there with their eyes closed or in
Okay.
light sleep stage, but not in REM sleep. And that's one of the big problems. But we're basically starting to analyze this data, and I'm reasonably optimistic.
So someone here has asked, is the RBD survey reliable? I don't know what that is, but presumably it's
So
some
the
question.
RBD, so there's about 5 or 6 validated surveys to make a diagnosis of
Yeah.
RBD. Very few of them have been tested in a community ascertain cohort, so that's
Okay.
the Mayo Sleep Questionnaire, there's a Hong Kong Sleep Questionnaire, there's an RBD screening questionnaire. General accuracy is around 80%.
Okay.
What the screening test does not do is rule out other sleep disorders like obstructive sleep apnea that will mimic RBD clinically and are
Mhm.
treated very differently.
Okay. So the way forward would be, I say, or probably not me, but my wife would say, I'm worried he's got RBD.
Yeah,
You would send us the kit. I wear it at home. You then get that information. You can
yeah.
analyze it. And then would that be enough to make the diagnosis, or would you still want people to come into hospital and have the sort of formal testing, or do you think actually we're moving to the point it's pretty reliable and that would do?
I think hopefully in 5 years I'll be more. I mean, they're already doing home PSG now in a lot of the
Yeah.
NHS
Yeah.
clinics, particularly for children, where that's very difficult to get them
Yeah.
in. And, I think in a selected subgroup, as long as the electrodes don't fall off and you've got good placement, good recording, it's probably going to be good enough. But I think there's no replacement for seeing someone who's a
Yeah.
sleep specialist or neurologist trained to say, look, this is what you've got, this is the risk or the implications for you. Here are the treatment options. What do you want me to do with you today? And do you want me to liaise with the GP, your family physician?
Okay. So just before we move on to the sort of final bit where I want to talk about sort of wearable technologies more generally,
Mm,
not just for RBD, just on a sort of practical, if you see someone in clinic and they've
mm,
got RBD,
Yeah.
what do you say to them? Do you
You're going to
say,
ask what I tell them. Yeah.
you've got problems down the road, or do you sort of say
Yeah.
there is a history of this? Because I see a few people with this, and they sort of say, well, is there, what's going to happen? And I'll say, well, it's very likely that over the next 10, 15 years, you'll transition into some state of the ones we've talked about, but I couldn't
So
tell you when. And I, and you know, and I, but I maybe I'm outdated on this. I mean, what do you say to
no,
patients?
I think there's no. I mean, I've just basically come to doing what I do now based on just seeing a lot of patients. And also, there's a number of very good surveys that have been done on people living with their RBD. Daniela Berg is one, others. And the majority wanted to know what the diagnosis was and what are the long-term implications for them. And I think they wanted that to be communicated in an easy, understandable way by somebody who knew what they're talking about and to know about that their lifelong risk of Parkinson's or future neurological conditions increases. So that
Mhm.
I generally start by asking people if they want to know, the implications of having this condition, and they always do. I
Yeah,
go into the risks, but then I talk about the positives. The positives are for me that we can then recruit them into research, which
yeah,
for an individual who's been diagnosed, they're not left dangling. We're going to be able
yeah.
to follow them up every year. We're going to be able to reassure them if they're sane, or if there is something there, we'll have a chat with them and suggest next steps and make that referral. And that's how I've done the ethics. I also encourage them about lifestyle. And I say for Parkinson's, we've got no neuroprotective treatments that slow down the progression other than exercise. And there's increasing evidence to suggest that that may slow it down. So I encourage them all to get involved with exercise.
Yeah.
And also, if they're on high blood, if they need treatment for vascular risk factors, I suggest that they get that treated. Um, so that's sort of roughly how I
So I
would
mean,
play that.
you basically tell them what if they would like to know the prognosis, what it actually means, and also what they could do
Mm.
about it. And obviously,
Yeah.
I guess going forward, as trials, and there are a lot of trials in POG at the moment, this will be an
Mm.
increasingly attractive group for doing
Yeah.
the sort of interventions, really. So coming on to the sort of last bit, Michelle. I mean, obviously they see you every year, you do your assessments, but
Mm.
we're quite good at assessments. But they're a bit subjective. Obviously if you see someone in
They
clinic,
are.
you examine them, you could be a bit tired, you could be a bit grumpy, they could be a bit tired, they
Mm.
could be a bit grumpy. So
Absolutely.
obviously moving towards a sort of wearable technologies for
Mm.
diagnosing
Yeah.
Parkinson's or tracking Parkinson's.
Yeah.
I mean, how far has that come? So would you diagnose someone with Parkinson's in your clinic based on a outcome from wearing some wearable
Well,
device?
yeah, interesting. Okay, so I got interested in a quick digital, low cost, pragmatic solution. And what I did is working over the last decade really with some clever mathematicians is to say, well, everybody's got a smartphone, whether it's an Android or an iPhone, and everyone can download an app. So let's design an app that really seeks to kind of in a more reproducible way, capture some of the really salient motor symptoms of Parkinson's. And so the app we do looks at speech because the patient has to go into the phone. And there's quite a lot there. We're not asking them to say words or read a poem.
No.
We're just looking at some of their features of their voice and their ability to maintain volume, the tremor. Then we measure their rest tremor and we measure their postural
And
tremor.
rest tremor by getting them
Um,
to hold the phone.
so rest tremor, they have to hold the phone on a hard surface, like a
Okay.
table, and then we get them
Okay.
to count backwards with their eyes closed. So that brings out a rest tremor. Before we just said, "Oh, put your hand on your leg," but then we found the leg tremor interfered with the arm tremor. So we say hard surface. We catch that right and left, and we do a postural tremor by just holding the phone out like this. We
Right.
also do a reaction time where they have to press on a button to a yellow square that turns up. And that's really important for attention. And attention and salience is, reaction times also affected by Parkinson's. And then we have a finger tapping test where they do this and
Right.
they do that right and left. And to me, that's when you see them fatigue is the best measure that we have of bradykinesia.
And do you do that? And
Lastly,
how's that done? Is that so the people you recruit to do that, do they
yeah,
do that every week? Do they
yeah.
do it every month, or do
Right.
they
So we
only do it
always
when they
try
come
and
to your
capture
clinic?
the in-clinic visit. We do
Yeah.
it because that's 8 minutes to add. But we also give people who are willing and like using a phone to take home. We use a Samsung and we ask them if they can do it 3 times or 4 times a day over a week, and then we get a courier to pick it up from their house. And
Okay.
we've been using that in the exenatide PD3 trial, which is a
Yeah.
phase 2 trial of a GLP-1 agonist. And the data for that and the outcome will be released, I believe, quite shortly in the next few months or weeks in the UK.
And have you used
So
that
we've been
device
using that.
outside of the trial? Do you
Yes.
think that so basically it looks quite useful as a way of tracking and
So
following?
yeah. So what we can do, I can diagnose Parkinson's with around 90% accuracy, simply based on the smartphone test alone. And we published that in Neurology.
Yeah.
No clinical information, nothing, just the smartphone. Areas under the curve between 85 to 93%
Okay.
based on the single test.
So
We've
you're
also
making us
shown the
redundant.
smart.
Nobody
Well,
needs to see us. They can just get their smartphone
yeah.
and
Yeah. Well, we do a lot more than that, though, don't we? But yeah. And then I'm testing it now in clinic, in my atypical Parkinson clinic, to look at whether it can distinguish early on people with other forms that are not Parkinson's. We then
yeah,
showed the app could predict the onset of meaningful events for patients. So milestones like the going from not gate freezing to gate freezing, not
yeah.
falling to falling, having no cognitive impairment to impairment in the next 18 months. So we
Okay.
published on that. And that's really useful because if you had a test that could predict
Yeah.
who was more likely to fall, you'd
You
probably
could
triage that patient to OT, maybe some physio.
yeah,
I forgot to
you
say
could put
that
in
the,
measures to protect them much more
yeah,
based on where things are going.
absolutely. And the app also has a standing test and a
Yeah.
gait test. It's like a timed up and go. So we measure
Okay.
most aspects. We can't capture rigidity because
And this
that
is
really
all on
means
the
that it's
phone.
all on the phone. And then lastly, we've been working on how well it can measure medication benefit by testing people off and on medication. We know from exenatide it definitely measures that, and we're looking at a single measure that the smartphone can predict that will be used for clinical trials.
So this is a beautiful way of using what everyone has without having to
Yeah,
go get yourself wired up or go and
yeah,
buy new gadgetry
totally.
and things like
And
that. Someone
I'm
wants to know if they can get the app. So can
Yeah,
they
so I was going
download
to
it?
talk about that. So we're providing it for a study called Transforming Parkinson's Care Across Africa. So we're providing it, we always provide it free of charge to our research collaborators. So there are 6 countries in Africa going to be using this over the next 5 years. We've collaborated with over 20 people at 20 sites. We now have just launched in the last month a spin out company called New Health. So I am what's called an academic founder member. I don't run that company, but I've basically made available that code and the test. But it's now also got memory, non-motor, medication, so the patient can use it as a reminder to take their tablets. That is linked to the recording, whether it's off or on. And we're now testing that in 4 PD clinics in the country. So you've
Okay.
got Norfolk and Norwich with one of your
Near us,
consultants.
yeah.
We've got Oxford. We've got the Hebrides, which are a
Oh,
very remote islands in Scotland, which are difficult, and you can understand telemedicine and its
yeah.
utility. And we've got one in East End, East London.
Okay.
So
It-
what we hope is to make the app more widely available because I got frustrated with not being able to offer it to
yeah.
patients.
Yeah.
And the New Health app, if you look at their platform, talks about the app. And my aim is to make that more widely available at really low cost for
Yeah.
individuals.
Okay. Well, I'm afraid we're pretty much out of time. I mean, it's been a fantastic journey, Michelle, from how you managed to resist doing law to going to medical school, fight
Mm.
against all of the obstacles which exist in medicine for women, for people having families. But you got through all that. You then set up this, fantastic cohort, which you've done the most amazing job with, and you keep the research moving. And now we're in this fantastic, exciting era where you can diagnose people before they've got overt Parkinson's. We're moving towards the fact this can all be done on a smartphone. And obviously, this will synergize very nicely with all these new trials that are coming along to modify it, either with drugs, as you say, with exenatide, or these vaccines and such like. So thank you very much. It's been a wonderfully inspiring and interesting conversation. I would just like to finish by thanking our sponsors, which are Supernus Pharmaceuticals and Mitsubishi Tanabe Pharma America. I want to remind everyone that this is not the last of the year. We have one more of these research spotlights, which will be on December the 3rd at the usual time, which is 4 o'clock in the UK, 11 o'clock Eastern time in the States. And that's with Malu Tanzi, who's from the University of Florida at the present time. and she's been talking about inflammation in Parkinson's, which is what Michele was also mentioning as being a key feature found in the blood with people with Parkinson's. So thank you very much for joining us this afternoon, this evening, this morning, wherever you are. Do not forget the conference. Michele, Eli has put up the poster for Phoenix, May 2026. So do go to that. There is nothing like it. It's absolutely fantastic. Tony Wilkinson has put one up from Ireland as well. So thank you all very much for joining. Thank you again, Michele, for a
Pleasure.
splendid hour going through your research and hearing about your life. So thank you very much, and thank you all very much for joining.